share my screen right away here and make sure that's all working. Um, okay, how does that look?
Your it's up looking great. Thank you.
Okay, great. Yeah, so thanks for having me. I'm a master's student, like Heidi said, at in the Juanes Lab at UVic, and I'm doing my master's degree on salmon diets. And so I've been working for the last couple of years on this project and just as a technician. And now I'm moving into being a student and we're going to be doing some more sort of analysis with it. But pretty much what we do is we collect the stomachs of salmon from anglers, from recreational fishers, and we dissect them and sort of get an idea for what they're eating. And we have a bunch of data that's associated with that and sort of there's a lot, it's surprisingly a large amount that you can learn from what these fish are eating. But anyways, I'll get into it here. So yeah, once again, a little bit about me. There's a nice photo of me showing, I've been a bit of a fish lover since I was a kid. But yeah, I've been working on the project since 2023 and excited to be for at least the next 2 years working as a master's student. So just a brief overview for what I'll be talking about tonight. So I'll just give a bit of a background on the program and then talk a little bit about comparing Chinook and coho salmon diets. So that's most of the data, or pretty much all the data we collect is on Chinook and coho salmon. And so looking at comparing those two diets, seeing how they are different, how they're the same, and what we can learn from that. And then I'll talk a little bit about historical diet data. And so there is some data available from the past that we can look at to see what fish we're eating sort of throughout time, and we can compare it to our own data to see how things maybe have stayed the same or how they've changed. And so we'll take a little look at that as well. So to start things off, we'll look a bit at the program. So this program was founded in 2017, and so we have about 8 years of data, but we have a few objectives associated with the program. First is to address, we have a couple short-term goals, and first one is to address basic knowledge gaps in Chinook and coho salmon diets in BC. And so I was talking a bit about that historical data that we have available. But the thing is, the most recently available data that we have is from the 1960s, apart from the data we're collecting. And so there's a pretty big gap there in the sort of the data knowledge that we have. And so we're trying to fill that through this program. Another goal is to improve our understanding of forage species ecology. So forage species are the things that salmon are feeding on. This is mostly fish in our case because Chinook and coho salmon are mainly piscivorous, which means they mostly eat other fish. So a lot of things like little herring or anchovies, squid are a forage species, that kind of thing. So we can learn about the things that they're eating by looking at when and where those fish are eating them. We also have some short-term goals with or sorry, that should say long-term, but we have some long-term goals with the program. The first is to develop and maintain a citizen science network. And so that is sort of our sampling process. We get um, all of our samples from citizen scientists, from anglers who are involved in what we do. And we use another long-term goal is to use salmon diets to monitor ecosystem change. And so as we gather more data over the years, we can kind of compare from year to year if things are shifting. Um, and, if they are shifting, if salmon are eating different things at different times in different places, what does that tell us and why might that be? And so we can kind of use it to monitor how those things are shifting. And one final long-term goal we have is to provide research opportunities for early career scientists. And so this program has employed a lot of people who have been coming out of their undergraduate degrees and looking for work, and it has help people like myself transition into programs like master's programs or to continue working in science and in fisheries somehow. So, all right, and now we'll get a bit into our methods and just how we do our sampling. So I already talked a bit about how we use private anglers for our sampling. And so we have this network of people that we work with, and they will collect these samples and then bring them into us. We also work with fishing charters, fishing guides. We'll sometimes attend fishing derbies where we'll pick up samples. And then we also work with some Creel surveyors, some First Nations groups, and a few NGO collaborators who help us collect samples. As part of this sort of sampling, we actually, for anglers, we provide some incentive prizes. So for every stomach you submit, you get your name put into a draw to win some fishing gear. So we have a few tackle stores and stuff that they donate fishing gear to us, and then we give it out to our anglers as sort of a thank you and a little incentive to participate. And I will say that if any of you out there are anglers and want to participate, we would love to have your fish stomachs. But yeah, you can let me know and we can get you set up with what you need. But once we have those stomachs, they get frozen and then we bring them into the lab and we do some processing. And so we take taxonomic identification. So we ID everything that they're eating as best we can. We take weights and lengths on all the prey items, and we also archive some physical samples. So you may or may not be familiar with something called otoliths. They are sort of the ear bones of the fish, and they grow in daily rings. And so this allows us, if we keep these little ear bones, it can allow us to tell how old the fish was, and things like that. We also collect some of the intestine contents for sort of more fine scale analysis. And then in terms of prey identification, a lot of the times in this middle photo here, you can see this fish is kind of missing its head. It's kind of tough to tell what it is. A lot of time it can be really challenging. And so we use this kind of a set of decision-making rules to identify the things that we're seeing. And we use a lot of both morphology and the hard parts of the prey items that we're seeing. So we'll get a little more into that. So you can see here are some photos of 3 different species of fish. These are all sort of the Clupeiforms group. They're anchovy and herring and sardine. And these are all things that we could potentially see around here. And they do look quite similar in terms of morphology. But when they're nice and intact like this, when they're not half digested, you can kind of tell the difference. The sardines have spots along the side that you can distinguish them. We also don't see sardines very often. But from these fish, we also get, so these are examples of otoliths, which I was talking about, so those little ear bones. And then this is another bone that sort of is where the gills attach into the head. And so you can see how for each of these species, each of those bones is slightly different. And so even if our samples look like this, or maybe like that with, it's all mushed up, or maybe it's just a bunch of goop inside of a stomach, we can still tell what was in there because we can look at those bones and yeah, ID things that way. So now I'm going to talk a bit about our data. This is the sampling distribution that we have for our Chinook salmon. And so each of the circles is a fishing site, and the size of the circle is the number of fish collected from there. And you can see it goes 10 to over 200 samples from a given site. And we also have the number of stomachs by month and by year. And so for our monthly samples, we get a lot more in the summer, and that's just because that's when recreational anglers are active. That's when people are out fishing, and so that's where we get samples from. But throughout the year, especially in this green, that's the Strait of Georgia, we get quite a few samples throughout the year, which is really interesting because then we can kind of have this long data set that spans almost the entire year. And then across years, we have pretty consistent sampling. The West Coast in pink is a little more inconsistent. We've had fewer samples in recent years, but overall, pretty good sampling. And I think about 5,000. We might actually be closer to 6,000 Chinook salmon stomachs that we've processed at this point. So a lot of data there. And there's a few ways that you can sort of analyze data and look at it. The way that we're going to be looking at today is just proportion data. And so we'll have just out of all of the things that are eaten, what percentage of that is made up of a specific prey type. And so we use the weight of those prey items. So say it's the weight of the herring out of the total weight of all of the fish that or all of the prey items that were eaten. So then you get proportions. There's a lot of other ways you can look at how full the stomachs are. You can calculate proportions in different ways, but this is kind of a simple way that's hopefully easy to understand for you guys. And so this is how our data looks. There's a lot of information here, so we're going to kind of go through things one at a time, but along each of the boxes goes around a different region. And so this map is back up here in the corner. And that is just showing those regions again with the colors. And so we have the Strait of Juan de Fuca, we have the Gulf Islands, we have Howe Sound, and we have the Strait of Georgia and the west coast of Vancouver Island. And then the top sort of row of those columns is the summer and the bottom is the winter. And then for each of those regions in each of those seasons, we have the each of the years we have samples for and the diet proportions within each. And so we'll talk about these one at a time, and we'll start with the Strait of Juan de Fuca. And so this is, I'm sure you guys are familiar with the Strait of Juan de Fuca, kind of between Canada and the States there. And on the side here, you can see the different prey types we have in the legend. And what you'll notice, especially throughout all of this, actually, you'll notice that that light blue color, which is herring, is super important in the diets. In the Strait of Juan de Fuca for Chinook salmon, there's a couple other things now and then, but herring's a big one for sure. There's, I think, one of our anglers with some big fish. Next, we've got Gulf Islands, Harrow Strait region. Once again, we've got quite a few herring here. This region also, we do get some sand lance, and so these are kind of like needlefish. Sometimes we have a stomach that's, this, I think this stomach had 400 sand lance in it, so they can eat a lot of them for sure. This next one is Howe Sound, so this is off of Vancouver. This area, interestingly, you'll notice this sort of dark blue color, and that is anchovy. And so anchovies are an interesting one because historically they haven't really been that present around this area, and they're more associated with like the coast of California. But in sort of the last 10 years, they've started moving in here and they spawn now in the Strait of Georgia, specifically around the Fraser River Delta and the Fraser River plume as it goes out into the ocean. And so that's why we see them in Howe Sound. And there's a picture of an anchovy. You can see they got a funny little nose. And looking at the Strait of Georgia, this area, we really see a really high importance of herring. And so I think people are becoming pretty aware of herring. There's a lot of attention given to herring right now. And part of the reason is because they're such an important prey item for so many different other animals in the Strait of Georgia. And that includes Chinook salmon. You can see these bars are mostly that light blue color. And that means that mostly herring is what's important in their diet. And then if we look at, oh, there's some photos of herrings that we've had in the stomachs. Sometimes we can get a whole bunch of them in there too. Sometimes they're a bit messed up, but these are some prettier looking ones. And then if we look at the west coast of Vancouver Island, we see this sort of dark red color that is cephalopods. And so that's mostly squid. You get these big balls of squid that show up on the west coast, and the salmon feed quite a bit on those. And so that's something we see pretty consistently. You can see across each of these bars contains squid, and that's sort of each of those years. It varies in importance, but it's present throughout. And so now I'm going to show the some of our data for coho salmon. And so this is our sampling distribution for coho. You can see those bubbles aren't quite as big. We don't have quite as many samples. And I think Chinook salmon are just more of a target of the fishery, and they are more of what we have kind of focused on. But if you look at this bottom plot here, especially in the last 2 years, in 2023 and 2024, we had some really big runs of coho salmon coming up into the Strait of Georgia and like record-breaking runs kind of thing. And so that is reflected in the number of coho salmon that got submitted to us as samples. And so that's really interesting because now, we can make these comparisons across years and see, yes, get some more sort of in-depth data on these coho salmon. The fishery for coho is closed during a lot of the year, and they're also not around during sometimes of the year. So we mostly only get kind of during those summer months, and it's not quite as consistent across seasons. And so now looking at some of this data, these are the same plots. We've got the regions are in those boxes, and then summer's on top, and then we've got winter. We only have winter samples for the Strait of Juan de Fuca, and winter is really just October and November and not any time after that. But yeah, we'll look at these ones quickly. So we've got in the Strait of Juan de Fuca, herring is still present, and so we've got this light blue color again. And so herring is definitely still an important diet item. These coho salmon still eat a lot of fish, but it's not quite as important, that the blue isn't making up quite as much of a proportion of the diets overall. We do see quite a few euphausiids, and so that pink color is euphausid, and so those are just krill. They're like little tiny shrimps kind of thing. But then also we get this sort of light green color showing up, and that's other or unidentified fish. And so mostly that'll be other fish species other than the ones we have listed here, which are anchovy, sandlance, and herring we have sort of in these plots. Those are the most common fish species we see for the most part. But then, yeah, and these light green bars will be made of a bunch of other fish species. And so that includes this photo I've got here is a juvenile salmon, which is interesting. And we'll talk a bit more about salmon eating salmon later. Yeah, and then we've got the winter in Juan de Fuca on the bottom there, similar kind of story. So yeah, now we can look at the Strait of Georgia. Herring, once again, pretty important in the Strait of Georgia, more important than sort of in those other regions. But for coho salmon, those herring are a bit less important. And so we're seeing that sort of red pink color there. That's other crustaceans. And so that is things like crab larvae. They float around in the water, or once again, euphausiids show up a little bit, but we also see amphipods, which are another sort of small planktonic crustacean. And so those are important in the Strait of Georgia. And then there's some photos of them. So on the left, we've got an amphipod, and on the right, we have a crab larvae. So that is probably a Dungeness or red rock crab. It's tough to say. I'm not an expert on that sort of identification, but yeah, that is what they look like when they're little babies. And if we look at the West Coast data, it's a little bit more variable year to year. And this is partially just due to the sampling is not super consistent across years, but we see herring and we see cephalopods, and it's kind of a mix from between different years. But yeah, definitely herring are consistently throughout there, and some of those crustaceans as well. And so now we can kind of make a comparison between Chinook salmon and coho salmon diets. And so if we're looking at Chinook salmon, overall, what we saw is that herring are most important. We're seeing those a lot. Anchovy are important in how sound, which is kind of an interesting one, like I was mentioning. And then we have sandlance in the Gulf Islands area. The Gulf Islands, and especially around like Sydney as well, up sort of Saanich area, North Saanich, the substrate there is quite sandy, and so the sandlance really like that area. They burrow in that sand and they live there. Um, and so that's kind of why we get those sandlance there when they emerge and come out of the sand. And then we get squid on the west coast of Vancouver Island. And so In total, we've had about 60 different prey types in over 6,000 stomachs that we've processed to date in the program. And so it's quite a lot. Most of those, once again, will be herring. And so every once in a while, we get a rare individual. But we'll compare that in a second to what we see in coho diets. And so in coho, herring are important, but they're a little bit less important than Chinook. We see a lot more crustaceans, things like euphausiids and those crab larvae and amphipods show up a lot more. Um, and we also see those other fish species, and we'll dig into that soon here. But in total, with coho diets, we've seen a total of 67 different prey types in under 2,000 stomachs. And so, it's a smaller sample size, and yet we've still seen more variety than we've seen in the Chinook diets. And so this is kind of bringing up an interesting difference we're seeing here that these coho salmon, their diets are quite a bit more variable. They're feeding on a lot of different things. And so to further sort of highlight that point, we can look at the other fish species that these coho are eating. And so here's some interesting photos. So first one is a Pacific sandfish. This was one, we only have ever had one stomach with Pacific sandfish in it, but it was like, there was like 6 of them in there. And you can see this photo below. I didn't take this photo, but it was taken from the internet somewhere. But they will bury in the sand and have their face sticking out, and then they're kind of a lion weight predator, and they'll jump up and grab things. But they're also, this one was pretty small. That one was only maybe 2 or 3 inches long. And so if the coho salmon is running into it, it'll eat it kind of thing. We also have had a prowfish, and so that's this guy in the middle there. It's a really interesting one. I wasn't on the program. I wasn't working when we found this fish, but super interesting thing to see. And then the third one is that we actually see quite a few salmon in the diets of these coho salmon. And that's interesting because it's cannibalism. And so cannibalism can have some pretty significant effects on sort of the survival of these species. If there's a lot of coho salmon and they're eating the baby salmon, then that could influence the survival of future generations. And we have seen pretty much all the salmon species being eaten by coho salmon. I think we've confidently ID'd other coho salmon, Chinook salmon, chum, and the pink salmon. I don't know if we've seen any sockeye that we can say confidently, but it doesn't seem like they distinguish between them when they're feeding on them. And so we can look a little bit more into this and comparing between Chinook and coho salmon diets, the adults. And we see from this plot, so we've got the different colors are the different regions again. You can see from the legend there. And then on the left, we have the Chinook salmon, and on the right we have coho salmon. And then the scale, so that y-axis going up and down here, is the likelihood that you will see a salmon in the diets of one of these salmon. So there's the likelihood you see like a little baby salmon that was eaten by an adult salmon. And it's a pretty distinct contrast here, where with these Chinook, you'll see all these points are really low. They're all really close to 0. We maybe see one every once in a while, but it's not very common. Whereas in the coho salmon over here, we're seeing, 10% in some regions. Like in Juan de Fuca, we've had close to 400 samples, and so that's a pretty good sample size. And still we're seeing, 10% of these fish will have a salmon in their diets, which is quite interesting. And so, yeah, like why is this happening? Why do coho salmon have such a more varied diet than the Chinook salmon? What are these differences the result of? Does anyone out there have any guesses on why they think their diets are different like this? I'll give you a second to think about it.
Mm. Oh gosh, that's a tough one. Mhm.
Yeah, no worries. Okay, well, I have a couple potential explanations. I wouldn't say these are like confidently what's going on necessarily, but I think it could be pointing us in the right direction. The first one is something called resource partitioning. And so this is something that we see sort of throughout nature, I guess, and throughout sort of predator interactions is that if everyone eats the same thing, then everyone's going to run out of food. And so, we have, I think, 5 species or 6 species of salmon on the West Coast. If they were all only eating herring, they might eat all the herring and then there's nothing left to eat. And so as these fish have evolved, they've learned to, some fish started specializing in eating more invertebrates, some fish specialized in eating more fish. And so they kind of diverge in this way so that they're not directly competing with each other for food sources. And so then there's sort of more overall food sources available to the different species. And so that's one kind of possible explanation. We can also think about habitat differences. And so coho are more of a near shore surface oriented fish. And so they'll tend to be in shallower water. It'll still be deep, like maybe up to 100 feet, but coho are also known to, surface strike, like you can fly fish for coho. They'll strike at a lure right at the surface, whereas Chinook are quite a bit deeper. You'll find them at 3, 4, 5, 600 feet deep. And so, but the thing is, these two things, this resource partitioning and habitat differences, could potentially, it's sort of a chicken and the egg thing. Like, which one came first? Did fish start eating different things because they were living in different places? Or were they already trying to eat different things and then the things they were eating were in different places and so they're following their food? And so I don't actually know that. That's a bit of it's probably not quite that simple. Um, but it's an interesting thing to think about, like what is sort of the driving forces that, make it so that these fish, they both live in different areas and they're feeding on different prey types. And so if we look, we pretend this is sort of our water column in the ocean here, and we have our depth from the surface down. Um, we have coho salmon and they're kind of at the surface there and they're feeding more on, small fish like other juvenile salmon or other juvenile fish and things like we got some little amphipods here and those, they aren't that deep in the water. They'll be like maybe in the top 100 feet of water kind of thing. Versus if we look at our Chinook salmon, they're a lot deeper. They're occurring more where those large herring are occurring, and so that's what they're feeding on kind of thing. And so yeah, there's this kind of relationship between where they are and what they're eating because that's where their food is. And so you might wonder about, well, what about those other species of salmon? We've got other ones. We don't research those personally, or with our program. Other species of salmon like pink and sockeye tend to feed a lot more on invertebrates. So coho salmon feed on those invertebrates, like those little crustaceans, but they're still feeding quite a bit on fish, whereas sockeye and pink salmon, they're mostly feeding on invertebrates. And that would be very miserable to dissect and do those stomach dissections. I won't lie to you, because when you get a stomach that's full of those invertebrates, you have to kind of count them all out and sort them all out. It become very tedious, and so we don't do those ones. We're also more interested in getting fish out of the stomachs because we're trying to learn about those fish as well. Um, so yeah, those also have different diets, and so they're kind of, there's more diversity in the diet, so they're not all overlapping completely. Chum salmon, they actually are quite interesting. They um, have sort of a larger stomach than the other fish, and they also have um, different finer gill rakers, I think, that allows them to eat things like jellyfish. And so jellyfish are not very nutritionally dense, like you're not getting a lot out of one jellyfish if you eat it. And so they have a bigger stomach that allows them to hold more food and get more nutrients out of it. Um, and that's from other research that's been done. We haven't seen that ourselves. Okay. Yeah, so that's kind of a comparison of our data with our Chinook data and our coho data. And now I'm going to go into a bit of a comparison of our data to some historical data that we have. Before I move on, are there any questions about the Chinook and coho salmon diet kind of stuff out there. No one's got any questions. I'll keep going, but feel free to put them in the chat or anything, or just turn on your camera or unmute yourself if you want. But yeah, so in terms of historical diets, like I said, the most recent data we have is from the 1960s, and so there's sort of this big gap between then and then when our program started in 2017, where there just wasn't any data. And so that was part of the inspiration to start our program, was to sort of address these gaps. And so today I'm going to look at a few of these publications that have this old data, and we'll make some comparisons to what we're seeing in our data nowadays. And so once again, this is our data. It's kind of organized in a slightly different way, and I organized it differently so that I can compare it to those historical data sets, because they just have sort of figures and stuff, and so we need to be able to compare it. But each of these sort of panels is a different region. We've got Juan de Fuca, and this is only data from the summer. And so this doesn't include winter diets. And so we have Juan de Fuca, the Gulf Islands, Howe Sound, and then on the bottom from left to right, we have Strait of Georgia, West Coast Vancouver Island, and some samples from Haida Gwaii. We only have 2 years from Haida Gwaii. We haven't looked at those yet. But yeah, overall, this is similar to what we were seeing before. Herring is pretty important consistently. Also, this is only Chinook salmon data that we're comparing to. But yeah, we're seeing herring, especially important in the Gulf Islands, that light blue color making up most of those bars. In Howe Sound, we've got anchovy, which is that dark blue, squid on the west coast of Vancouver Island. Oh, sorry, Strait of Georgia was down here. But yeah, so we kind of already went through a little bit of that, but just to refresh you on what that looked like, and then we'll make some comparisons here now. So the first one we have is this guy Williamson in 1930 published this paper. And this is they didn't actually publish data, they just had observations. So this guy was just out there, he was cutting open stomachs, taking a look, just kind of writing down what he saw. So they don't have numbers, but they have descriptions. And this is from 1926 to 1929. And so this is almost 100 years ago that they were looking at this. And some of the highlights from this, they saw a really high importance of sand lance. And so we've seen a little bit of sand lance in our data. This is from the West Coast. We do see a little bit. I'll throw up a plot here of our data from the West Coast. That dark blue color is sand lance. And so we see it a bit, but back then in 1926, sand lance was a lot more important, it sounds like, as well as euphausiids, which we see a little bit of, but not that much. In their data, herring and cephalopods were present, but they weren't particularly common. And cephalopods and herring are the most important diet items in our data. Yeah, in our data. And so I'm not 100% sure why these descriptions they gave us aren't very detailed, and so it's tough to say. It could be that where they were sampling was not similar to where we were sampling, and that's why they were getting diet differences. But there could be other things going on with, these populations of herring and cephalopods having different distributions back in those days. And now we'll look at another paper here. And so this is published in 1936. And so this is from Swiftsure Bank. And so Swiftsure is, if you can see this little red box here, that is that fishing site. It's kind of off close to Port Renfrew there. And so, yeah, this is data just from Swiftsure Bank. And so for this comparison, I've just directly compared it to any data we had from that same location. And so we had a bunch of samples from 2022. That's this bar here, compared to that 1936 data that was from the historical data. And there's a pretty striking difference here. And that is this dark, sort of bright blue color, and that is sardine. And so back in 1936, sardine were a very important diet item for these fish, and we'll see this come up a couple more times. Oh, actually, I think that was something I forgot to mention from the previous one, is that sardine were also quite an important diet item in these observations. And we'll see sardine come up a few times, especially for the West Coast. And so, from 1926, sardine was important. From this 1936 one, we're seeing this bright blue. Sardine is quite important versus nowadays, more recent data, we barely ever see sardine. I think we've seen maybe 8 sardine total in all of those like 6,000 stomachs we have. And so pretty uncommon now. And we'll talk a bit more about that later. But here's another paper. So this one's from 1944, and this data was from 1939 to 1942. And this research also used recreational anglers. And so they actually paid anglers 10 cents a stomach, which I think back then was probably a pretty good price, to collect these samples for them. And so this is just showing, once again, their sampling. Their map is a little hard to see. So in these red boxes is all the areas where they've gotten samples from. And it's pretty impressive how extensive they were able to fish all around the island back in 1939. And that's just a comparison to our data again. And so now if we look at the data, so each of these panels is a different region. And then we've got the historical data, we've got the years sort of on the bottom there, and then there's a gap, and then the gap is followed by our data. And so those gaps between the bars kind of show, yeah, just comparing the historical data to our data. And so if we look at Haida Gwaii, on the first one, we see quite a few more sandlats back in those early days compared to nowadays. In the Strait of Georgia, once again in the 1940, 1941, sardine appeared to be really important, especially in 1940. We're getting that bright blue color versus nowadays in the Strait of Georgia, consistently throughout years, we're not really seeing sardine. Herring is the main thing we're seeing. On the West Coast, no sardine in 1939, but then in 40 and 41, we saw sardine show up. And so they kind of have this fluctuation. But interestingly, no cephalopods again. But we're seeing cephalopods, like these are like squid on in more of our data. And we've got a couple more here. And so this next one's from 1957. And so once again, the gap separates the 1957 data from the more modern data. In this one, you'll see in the Strait of Georgia, that light blue color is smaller. And so it appears that herring was less important in 1957 or wasn't as available to those Chinook salmon compared to what we see nowadays. Versus on the West Coast, we're seeing a lot more sandlats and also quite a few herring. And the herring is interesting because if we now look at another figure, and so this is from the Department of Fisheries and Oceans, and this is their record of the spawning biomass of herring. And so this is kind of saying about how many herring were there in these areas. And so this top line shows the spawning of herring in the Strait of Georgia, and the bottom one spawning in the West Coast. And this red box here is highlighting 1957. And so if we look at 1957, that in the Strait of Georgia, the herring spawn was pretty low, but on the West Coast, the herring spawn was pretty large. And then if we look back at this old data again, Strait of Georgia, not very many herring. And so where that herring spawn was low, we're not seeing as many herring in the stomachs versus on the West Coast, there was more herring spawning and we're seeing more herring in the stomachs. And so it's a pretty, I mean, we don't have like concrete evidence to show that this is what is causing this, but it's a pretty interesting pattern that we're seeing here. And so if we look at this last one here, this is from the Strait of Juan de Fuca. And in this one, quite a low amount of herring, like surprisingly low compared to our data, that light blue color again. And it kind of was replaced more by euphausiids in this pink color and sandlats in the dark blue color there. And so in this one, if we look at that herring spawning again, so now we're looking at 1967, 1968. And so that was when the data for that last one was from, 1967, 1968. And we see that for both the Strait of Georgia and the West Coast Vancouver Island, that spawning index was quite low. And so, the Strait of Juan de Fuca positioned right in between the West Coast and the Strait of Georgia. Um, and so, neither of those areas had very good herring spawning. And so that probably translates to Strait of Juan de Fuca. There weren't very many herring. And so then we're seeing fewer herring in the diets. And so these salmon are being forced to switch to different prey types. And that could potentially be important because fish like herring have a lot of energy per fish versus things like euphausiids and like little crustaceans and krill. They are not as quite dense in the energy that they have. And so you have to eat more of them to get the same sort of energy content. And so this has, potential for the implications for these salmon survival. If they're not able to get that high energy food like herring, um, they might not be able to spawn as well. They might not be able to swim up their rivers to their spawning grounds. They might die before they can get there. But yeah, so these are some, potential implications if these herring populations are shifting. And nowadays, there's a lot of focus on herring, like I was saying, because they are so important. And so, if we were to see herring populations crash, that could have big implications for these salmon. Um, and so this is just an overview looking at all of the historical data compared to our data. And the one that I want to point out again is that sardine. And so this bright blue color, if you can see where my mouse is there, we're seeing it quite a bit in 1936 and 1940, 1941 on the West Coast, and then 1940, 1941 in the Strait of Georgia as well. And if you remember, there was that paper from 1926 where they described the diets and they said there were quite a few sardine there. And so this is an interesting one because sardine are very similar to herring. They're also a very high energy food source, but they're one that we're not really seeing these days anymore. And so if these sardine are disappearing, that's a food source these fish don't have. It might affect their ability to, survive and to spawn and to, create the next generation kind of thing. And so what could be going on with these sardine? Well, sardine are typically associated with the West Coast of California. And so they spawn in sort of warmer waters usually, and then they'll migrate north, sometimes as far as Alaska if the conditions are right. And so that's usually more warm water conditions, they'll come up north. And the populations have definitely fluctuated. And so, in our data, we see sometimes there were a bunch of sardine in those historical data sets. Sometimes there weren't as many, but they were typically present, especially off the West Coast. And the sardine kind of disappeared in sort of the late 60s. We didn't see them for a while, and then they reappeared in the 90s and 2000s. And off the west coast of Vancouver Island, there was actually a sardine fishery. And so they were quite abundant. And they fished them, I think, until 2012. And then suddenly in 2012, or maybe 2013, there just weren't any more sardine there. And then 2 years later, 2014, the sardine population crashed off the west coast of California. And so, and this is despite the fact that, these sardine are associated with warm water conditions. 2014 was warm water conditions, but we still weren't seeing these sardines. And so this population crash, down in California, it was affecting these ecosystems all the way up the coast, all the way up to where we are. And these fish, unfortunately, we don't have data for those years to see if they were important to Chinook salmon at that time. But based on the historical data and what we're looking at, we can only guess that, if they were present, they were probably important. And so now we're seeing them disappear. And so once again, similar to herring, this population has disappeared. This could affect the survival of Chinook salmon. And so that just kind of highlights this sort of ecosystem, how broad the ecosystem effects of something like, sardine from California can affect things all the way up the coast, which is interesting to consider. And it's also highlights the importance of, considering that in management, if we're managing things, we kind of often will have to think across borders in order to do it effectively. And so a few conclusions. First, we looked at Chinook and coho salmon diets, and they were able to teach us a bit about their behavior, right? And so we saw that coho salmon were shallower, and they were feeding more on different species that tend to be shallower. They have more variety in their diet versus Chinook salmon were deeper. They were feeding on those deeper fish, those larger herring, that sort of thing. And then comparing to historical data allows us to see how sort of those prey items have changed, how prey distribution has changed. And yeah, it can give us some real insight into how that may affect those Chinook salmon if things change in the future. And for future directions for myself, so I'm a master's student. I just started this semester, but we're planning on doing some fancy spatiotemporal modeling. So in other words, we're going to be making some cool maps that show us exactly where and when those different prey types that we have in the diets are important. And so I'm looking forward to sort of getting into that more, getting into a bit more of the statistics and modeling stuff. And so we'll be able to sort of get a more detailed look at the different patterns in what those fish are eating. And we're also hoping and planning on continuing with the program. I think we have funding for another year or year and a half or so, maybe 2 years. But yeah, we're planning on continuing to do this monitoring, and hopefully, it would allow us to, if things start to shift, if, maybe herring start to disappear, or if there's another fish species that starts to be more important, we'll see that in our diets and we'll be able to sort of track how these ecosystems are shifting, which is interesting and also important for, protecting these salmon because they need good, healthy food sources in order to survive. And yeah, and Chinook salmon, they're a really important prey item for the resident killer whales. And killer whales have, really big focus on them right now, their conservation. And those southern residents feed primarily on Chinook salmon. And so, if we're going to protect killer whales, we got to make sure there's enough Chinook salmon for them to eat. And if we want there to be enough Chinook salmon around, we need to make sure that there's food for them as well. And so, yeah, it's all kind of connected and important to keep in mind, I guess. But yeah, and here's a few of our sponsors and partners in what we do, primarily funded by the Pacific Salmon Foundation. So we really appreciate their support. And that is all I have. And so if you have any questions, feel free to ask. And thank you so much for listening.
Awesome job. Thank you so much. Yeah, you're getting a few claps here. If anyone wants to, you can ask Nathaniel some questions. So you can either put them in the chat and I'm happy to read them out for you, or you're welcome to raise your hand, turn off your mic, or turn on your camera, turn your mic back on, and you can ask the questions yourself directly. I'll just read out to you. I see in the chat already, David Rydell put in, I was about to say the dependence on Chinook of high energy herring would seem to explain why the health of the resident killer whales also depends on Chinook. As the saying goes, you are what you eat, which seems to apply to Chinook and resident orcas. So yeah, I guess
Yeah, for
important
sure.
part of that story. Mhm.
Yeah. Yeah, the energy density thing is definitely important. And like that is, one of the theories for those southern resident killer whales is that, Chinook salmon sort of anecdotally, they used to be a lot bigger. Like you see those old, I think I had a photo in my slideshow of this guy with this huge Chinook salmon, right? And so if these killer whales are able to get these really big fish, they only have to eat a few of them to get a good meal versus if the Chinook salmon aren't growing as big, they're going to have to eat a lot more of them, a lot more energy expended to get sort of less food kind of thing. And so, yeah, it is a really interesting relationship and how that sort of scales all the way down the food chain. Like if you have lower energy food that the Chinook are eating, then the Chinook are going to have less energy and there's less available to those killer whales.
Well, other people think of questions. I sort of had one, but I think what I was going to start asking you about is if you were able to see any kind of change in diet with the size of the salmon, but I guess you're kind of limited to a certain age because you're getting mostly catch, or did
Yeah.
you guys look at that, or?
So there's another project that's separate from this in the lab I work in that looks at juvenile fish. And so that's a really big, that is a really big like focus in general, in sort of the science research community. Because yeah, we're looking at adult salmon specifically. But when fish are small, the one of the key points in the life of a juvenile salmon is when it's able to get big enough to switch from eating those little invertebrates and stuff to eating fish. Because like I said, those fish are more energy dense. And generally speaking, a fish is limited in what it can eat by the size of its mouth, by its gape width. And so once a fish gets big enough that it can fit, a little baby, like a little baby herring in its mouth, all of a sudden it's going to switch to eating herring because those are way better food source. And so that is sort of a thought, that could be one of the sort of like, I guess you could say, bottlenecks of, for these salmon survival, is that as they're growing and as they come out of these rivers, they need to get to that size so that they're able to start feeding on those little herring, and those little fish that are available to them. And if they can't grow fast enough, then those herring are also growing, and so they might get sort of outgrown where they have to keep eating the invertebrates, and then yeah, and so yeah, definitely they when they're younger, they eat more invertebrates, and then as they get older, they switch. And I think the bigger the fish, the more they tend to eat fish.
Hmm, very cool. Um, I really like the story of like people are already catching kind of salmon anyway, so let their stomach contents to tell the story. And what's really cool is I've heard that herring are doing really well, especially in the Strait of Georgia in the past little bit, and I love that we're seeing this reflected in the diet.
Hmm.
Is there, I think in the Haida Gwaii region though, this herring aren't doing as like much of a boom, but you guys don't get stomach contents from up there, right? Is that just because of distance or?
Yeah, it's a bit of a logistic thing. Like I think
Mhm.
we had 2 years where we did get Haida Gwaii samples. But it is just like getting those stomachs frozen and then transferred all the way down to Victoria because our lab's in Victoria. It's a lot to sort of coordinate, and so we mostly focus around sort of the southern Vancouver Island. Um, but yeah, definitely cool to see that, herring have been the spawns have been quite large in recent years, and then we're seeing more of those herring. Um, it is interesting because we get sort of a variety of different ages of herring in the stomach, and so um a lot like historically people have kind of treated herring as if they all migrate during the summer out to the west coast of the island, because that's where they tend to feed. But what we're seeing is that there are quite a few adult herring that actually stay in the Strait of Georgia in the summer, that are being eaten by these Chinook, and they're quite important to them. Yeah, and there's a whole nother research project going on that topic, and so that's a whole nother like can of worms or can of fish, I guess. But um,
Does anyone else have any questions they'd like to ask? You getting some nice comments in the chat?
yeah, just looking at them now. Thanks, everyone. Thanks for joining. Glad you
Mhm.
enjoyed it.
Um, yeah. Yeah, it's been very interesting. Thank you so much for your time. I guess the last thing that I just would say is, yeah, thank you also to our audience for coming out. Sounds like you all were super engaged, which I really appreciate, and a small but mighty audience. This is actually the last marine night for quite a few months. So if you guys have any feedback about these sorts of things, I'd actually encourage you to reach out to the Victoria Natural History Society. I'm just going to put one person in the chat right now that, you know, is someone that you could reach out about either ideas of how we can improve this or talks you'd like to see featured moving forward, that sort of thing. But yeah, mainly it's made possible by great audiences like you, and then people like Nathanael donating their time to create really cool stories and tell us great things. So thank you to everyone. I hope you have a great rest of your Monday night, and perfect timing. We are ending like right at 8:30. So this was incredible.
Perfect.
Yeah. Okay. Thanks again and take care. Good night,
Great.
everyone.
Thank you.
Thank you.
Bye.